physiology lab assignment on carbohydrates and the pathway of carbohydrate digestion and absorption. So, do you ever start salivating before a long-awaited beignet? Well, this is actually your body preparing to eat. When your body sees or smells a beignet, it sends a signal from your sensory receptors in the oral cavity to the brainstem, which is then sent to the salivary gland to initiate the production of saliva. So, saliva serves many functions. It helps with the breakdown of starch by the function of salivary amylase. It helps with chewing and swallowing. It has a lubricating effect, and it also has a cleaning effect to wash away food debris and bacteria remaining in the mouth, and it basically helps facilitate the diffusion of food into your stomach. Um, so this is a little overview of the carbohydrate digestion. So the process of the breakdown of starch begins in the mouth, where the salivary amylase breaks down starch into shorter segments, which are called oligosaccharides. It functions best at a pH of about 7, which is that of the mouth, so when it gets to the acidic stomach, it denatures very quickly. Being a protein, amylase is then digested by pepsin. About 50% of dietary starch is digested before it reaches the small intestine. Digestion resumes in the small intestine when chyme mixes with the pancreatic amylase. The digestion is completed when the chyme contacts the brush order of the enterocytes. The two brush order enzymes, which are dextranase and glucoamylase, along with maltase, make glucose, which is then absorbed. The main tissues used are salivary glands and the pancreas, and the brush border of small intestines. The main hormones that are responsible for facilitation are insulin and glucagon, which is on here. And then glucagon is also produced by the pancreas, and results in the release of liver glycogen stores in order to maintain blood glucose levels during fasting. Insulin is secreted by the pancreas and helps with uptake of glucose in the cells for energy metabolism. Some of the factors associated with absorption include monosaccharides, which are absorbed across epithelial cells of the small intestine into the bloodstream down the concentration gradient. The main transporter is the SGLT, which you can see right here in this diagram, which is a sodium glucose transformer. Another thing that I thought was pretty interesting was, so this shows about how the SGLT also absorbs galactose, whereas fructose is absorbed by facilitated diffusion using a separate carrier that does not depend on sodium. Another thing that I thought was very cool was learning about lactose intolerance. So I learned that typically mammals are unable to drink after the age of 4 because of not having the enzyme lactase, which is used to digest lactose. But this is not the case in humans. I also thought it was interesting how there is a racial divide, and mostly white people are able to drink milk after the age of 4 because they have relied on a diet of cattle, and use milk. And so the mutation for being able to drink milk is on the chromosome 2 called the LCT gene. And in most people about the age of 4, the MCM6 gene shuts down the LCT gene, which is they're both on the same chromosome, chromosome 2. But people who are able to consume milk without any problem have the mutation in the MCM6 gene, which makes it unable to shut down the LCT gene, and does not seize the lactose, and it makes it able to digest lactose.